Hello students, welcome to chapter 3. We're going to be talking about biological molecules, basically talking about the different biological molecules, the nucleic acids, the carbohydrates, lipids, stuff like that. Okay? Formation and modification of biological molecules. Nearly all organic compounds have to contain those carbon-hydrogen bonds. So we kind of talked about this during lab. Organic specifically means that it has to contain carbon, but it also has to have those hydrogen bonds involved. So things like methane, table sugar, DNA, things like that are organic, right? Anything like table salt or silver, even carbon dioxide, those are inorganic molecules. Even though carbon dioxide has carbon in it, it doesn't have a hydrogen bond attached to it. So organic molecules, these have 4 major classes, right? Carbohydrates, lipids, proteins, and nucleic acids. These are found in living components, living organisms, right? So consists of hydrogen atoms, hydrocarbons, right? Hydrocarbons. So we have simple hydrocarbons that have single bonds, double bonds, or even triple bonds, right? You can have like methane, ethane, different ones like that. Or you can have chained hydrocarbons, where you have those hydrocarbon chains. Sometimes they can be in like a zigzag formation, where you have like octane or even nonane. The simplest hydrocarbon is methane. That's CH4. That's 1 carbon attached to 4 hydrogens. Sometimes you have a hydrocarbon ring, right? So down there at the bottom you have benzene. That's 6 carbon carbohydrate, and it just forms a circle, right? But all hydrocarbons can form a linear and a ring hydrocarbon, depending on how long it is. So we talked about functional groups in labs. Functional groups are these small reactive groups that attach to those R groups. Those R groups can be the hydrocarbons or whatever is attached to it. So those are those engines that run the system. So we have different types of functional groups: hydroxyl, carbonyl, carboxyl, amino. Different ones we didn't talk about are phosphate and sulfhydryl. I think there are some other ones, but these are some ones that we can talk about. Functional groups are linked by covalent bonds, right? So all of these organic molecules, all of them are covalently bonded, represented by the selective symbol R. So that R sign is covalently bonded to probably a hydrocarbon or a another type of biological molecule. Isomers. So whenever we talk about molecules, sometimes these molecules can have different shapes. And two forms of the same chemical formula can have different chemical structures depending on how they're formed. So you'll have the exact same empirical formula, but they'll look the exact opposite. They'll look mirror opposite of each other, or they'll look completely different from each other depending on where those atoms are in the molecule, where those elements are. So stereoisomers are just the mirror images, right? So glyceraldehyde has two different forms. There's an L form and a D form, right? So the L form has the hydrogen on one side and the R group on the other side. And then the D form has it completely opposite. And this may change the way that it reacts depending on how enzymes form, how the molecules react to other molecules, how it enters reaction. Okay, so structural isomers, this goes into kind of what we talked about during lab. Structural isomers are two different isomers that have the exact same empirical formula, the exact same chemical formula, but they're arranged in a different way. So it's kind of like where the aldehyde and the ketone, right, where that CO group, that functional group, that carbonyl, has changed places. See that on the side there? It's the exact same chemical formula, but that carbonyl group has changed places depending on what type of molecule it is. And once again, this can change the way that this is, the enzymes are formed, the way that the chemical bond is formed in either the way you digest it or either the way that the enzymes break it apart. And one thing that we talked about also during lab is dehydration synthesis. This is a very important step in making polymers, making large molecules, macromolecules. Okay, carbohydrates, lipids, proteins, nucleic acids, these are all able to make polymers, able to make large molecules into a chain, right? And this is done through dehydration synthesis. So this is also called polymerization, right? By dehydration synthesis. So what happens is you have two different subunits, either a carbohydrate, a lipid, a protein, or a nucleic acid, and you have two different sides. One has an OH end and one has a hydrogen end, right? And then you have one takes off that hydroxyl end, one takes off that hydrogen end, and then you bind what's left, right? And then what do you have? You bind the two separate subunits together, and then you get water floating off. And you can see that on the side here. Dehydration synthesis reaction and water. You get an oxygen bond and water. And it just chains. going back and forth, back and forth, back and forth. Now the breakdown of this is called hydrolysis reaction, right? Hydrolysis is just the exact opposite. So hydrolysis meaning that water is breaking. Lysis means breaking, right? Hydro meaning water, water breaking. So water is coming in and breaking those two joined subunits apart. So hydrolysis meaning water is breaking those subunits apart. So the exact same, or the exact opposite reaction. And you can see it right over here on the slide. Okay, now that we talked about how these macromolecules are formed, let's just talk about the actual macromolecules. So, carbohydrates. Carbohydrates contain carbon, hydrogen, and oxygen. That's it. Right? These are simple sugars that can form into long polymers, right? Monosaccharides, those are the simple sugars, right? They can contain 3 to 7 carbon atoms. Monosaccharide of 3 atoms is triose, and then you have glucose, which has 6, pentose, which has 5. You have different ones that has a different amount of carbon atoms. When you combine 2 monosaccharides together, if you combine those using the dehydration synthesis, if you have 2 of them, that's disaccharide, right? Anything more than a disaccharide is a polysaccharide. So if we talk about monosaccharides, first of all, monosaccharides, soluble in water, sweet tasting, right? Any type of sugar that you mix in water is going to be sweet tasting. It's going to be like a fruit, right? Any type of fruit that you eat is going to have a monosaccharide in. These are things like fructose, glucose, triose. All of those are monosaccharides. Anything with the ending -ose in it is going to be a saccharide. All monosaccharides can occur in linear form. All right? Monosaccharides with 5 or more carbons can have a ring form. It can fold back on itself into a ring form. Now, it doesn't always do that, right? It does have that capability and it is more structurally sound that way, but it doesn't always do that. Again, we have disaccharides, right? Disaccharides are formed by dehydration synthesis of 2 monosaccharides, right? We have 2 monosaccharides, dehydration synthesis comes through, 1 water goes away, right? Maltose is a form of disaccharide. Maltose is a form of 2 glucose put together. Sucrose is formed by glucose and fructose. Then lactose is formed by galactose and glucose. Okay? Try to remember that. Maltose is formed by a glycosidic bond, right? Glycosidic bond just means that you have an alpha 1 to 4 linkage. Okay? This just means that you have a 1 to 4, that just means the carbon first and the carbon fourth. Carbon prime 4 and carbon prime 1, that just indicates what carbon it is. okay? It's very specific in what type of carbon it is, because your body doesn't react to the same type of chemical depending on how it's formed. Okay? If it's an alpha 1 to 2 linkage, it's sucrose. See that? So it'll react to that differently depending on also if it's glucose and fructose. And then if it's galactose and glucose, it'll be a beta 1 to 4 linkage. So it'll bond differently depending on what type of sugar it is. And then we get into polysaccharides. So polysaccharides, the most common polysaccharide are starches, glycogen, and cellulose. These polymers of 100,000 glycose units. So you can have chitin. Chitin is a type of bug polysaccharide, or you can also say animal polysaccharide, that assembles glucose units with addition of nitrogen. That's actually the exoskeleton of insects. Polysaccharides, these may be linear or unbranched. They also can occur in branched form. Alpha 1 to 4 linkage can form up starch granules, right? They can form up these amylose grains and then put them into these vacuoles, right? And then store just a lot of starch in there. But then you have beta 1 to 4 linkage, right? Glucose cellulose units, which stores cellulose. So we can digest some forms of starch, but we can't digest cellulose. And it's all because of that flip in that alpha beta linkage. And it's quite interesting because all it takes is that flip in that chemical formula and we can't digest it anymore. We're unable to digest cellulose even though it's the exact same chemical formula. Lipids. Lipids are water insoluble. These are non-polar biological molecules. Again, composed of hydrocarbons. Neutrolipids are stored, used as energy source, right? We store them in our bodies as energy source. Some of us, especially me, store a little bit too much. Neutrolipids are stored and used as an energy source like oils and fats, right? Fatty acids contain single hydrocarbon chains with a carboxyl group. And then you have phospholipids, right? These are in your cell membranes. We kind of talked about this before. Every cell membrane that you have has those phospholipids in them. And then you have steroids. Steroids are your cholesterols, are your sex steroids like testosterone, estrogen. Those are all lipids. So first off, let's talk about the neutral lipids. So glycerol, triglyceride, these are all types of neutral lipids. These are formed by dehydration synthesis. You have 3-carbon glycerol with an alcohol on it, right? That OH bond, that hydroxyl bond on it. And then you have some fatty acids on the end. And then what happens is you have the glycerol and those 3 fatty acids. They'll come through, and the fatty acids will have those OHs on the end. They'll take off the OH, and the glycerol will take off 3 H's, and then they'll bind. All 3 of those will bind together. So you'll have 3 forms of dehydration synthesis forming one triglyceride. That's why it makes it tri. So overall, the polar groups of the glycerol are completely eliminated because you're getting rid of that OH bond on either side. You're getting rid of the polar ends. So you're forming basically a non-polar triglyceride. These functions of the triglyceride, they serve as energy reserves, right? Underneath your skin, the fatty tissues right underneath the skin, they serve as the energy reserve that's twice the calories per gram as carbohydrates. So depending on how much carbohydrates you actually take in, that's twice as much carbohydrate, or twice as much calories as you could take in per gram. Of course, you have to then be able to get it out, right? So you have to burn that off through exercise and everything. So in mammals and birds and other things, this layer of fatty tissue is used as insulation, right? So in the upper climates, the upper latitudes, this is a layer of insulation for, those mammals and birds and such. Birds also use this as waterproofing. Ducks that have a special gland inside their cheeks that they'll preen their feathers with these triglycerides so that it is waterproofing it. Another neutral fat is fatty acids. So saturated fatty acids and unsaturated fatty acids. Saturated fatty acids binds the maximum number of hydrogen bonds, right? So if you take the triglyceride and you take off that glycerol, you are left with just the fatty acids, right? So the fatty acid is just the straight, or the saturated fatty acid is just the straight fatty acid. You only have single bonds, right? No double bonds exist in this. This is found in fats such as butters, solid animal fats. These are more readily digestible. You can break those single bonds really easily. You can, enzymes can break those pretty easy. So if you have one double bond, you have monounsaturated. If you have more than one double bond, you're called polyunsaturated. So if you have one double bond, you have a kink in your system, in your line, and that's what you see on the side here, right? One double bond gives it a kink. If you have two, you're gonna have multiple kinks, or more than one, you have multiple kinks. So unsaturated fatty acids, these things like any type of plant oils or vegetable oils, things like that, they're gonna have kinks in their system, in their lines. And whenever you eat vegetable oils or other things like that, it's very hard to digest that, right? And it's gonna bog down your system. So what happens is commercially converted saturated, unsaturated fats have to go through a process called hydrogenation to try and convert those into saturated fats. So if you ever go to the store and like get butters with unsaturated fatty acids, and it says something like made from canola oil or made from vegetable oil, yeah, that's because it's gone through a process called hydrogenation. You have to put it through hydrogenation in order for it to do that, because plant oils aren't naturally saturated. Now we're going to talk about waxes. So these are fatty acids with long chain alcohols. These help protect against water. These are weatherproofing. Also lubrication, pliability, things like that. It's like a waxy coating, right? So our nails kind of have a waxy coating on them, but a lot of leaves have this. So if you ever see the shiny part on the top of a leaf, that's a wax. It's a very shiny wax. And this over here, this is called a lotus plant. Now, if you've ever seen a lotus plant, or if you've ever come in contact with one, and you rub, or if you spray water on top of a lotus plant, lotus leaf, it'll bead up and it'll wash off almost immediately. That's because it has these very, very thin wire-like wax tubules. Very, very, very small wire-like wax tubules. And it'll just bead any type of water, any type of liquid on top of it. And this is actually where the weatherproofing spray substance, where they got the idea from. And this is an evolutionary trait, because these lotus plants live in water. And they don't want to be bogged down by the amount of water. Otherwise, they can't get the sunlight, they can't get the natural sunlight to their leaves. All right, next we're going to talk about phospholipids. So we're going out of neutral lipids, we're going to talk about phospholipids. So these are phosphate-containing lipids, right? And these have different parts, different structural parts to them. First, you're going to have a polar unit, right? The first topmost part is going to have a polar unit. And that polar unit is going to consist of a amino head to keep it polar within the aqueous solution. That's to keep it upright within the aqueous solution. If you think about it, where is these phospholipids located? It's going to be located within a border around your cells, right? So all of those heads are going to be all around the outside right, of that aqueous solution, so all around the water. And you want polar to be touching polar. So that amine group, that amino group, is a polar head that's touching the water on the outside. Next comes the phosphate group. And that phosphate group binds with the structural polar head. Okay. That phosphate group is just there as a binder between that and the glycerol, which is at the bottom. And that glycerol binds two fatty acid chains that comes down. And remember what I said about the fatty acid chains between the triglycerol. They're both nonpolar. So nonpolar is going to mix with nonpolar so that they can keep each other within line. Right? So polar is going to mix with polar is going to mix with polar, and nonpolar is going to mix with nonpolar, so that it keeps a wall between the outside and the inside. It's actually quite an ingenious method, and it's just all throughout chance through evolution. It's all molecular evolution. So hydrophilic heads on the outside, hydrophobic tails on the inside. And it's called the phospholipid bilayer. The film of phospholipids, 2 molecules thick. Structural basis of all the membranes in life. All right, next we're going to talk about steroids. Steroids are important because they also are contained within those bilayers, and we can talk about that the next chapter. I think it's the next chapter, either that or the following chapter. But steroids are important, especially because they have a lot to do with your body and a lot to do with your systems. So steroids are lipids with structures based on a framework of four carbon rings. So you're going to have different rings. You're going to have 4 of them, right? All of these carbon rings are going to be put together. So sterols, the most common type of steroid, will have a single polar OH group linked to one end of the ring framework. You'll have a complex non-polar hydrocarbon chain at the other end. So sterol, anything with the word sterol, like cholesterol, that'll have an OH group, and then you'll have some type of complex hydrocarbon chain at the other end. Right? So you can see that on this slide. Any type of steroid hormone will control development or behavior, anything like that. Right? Like testosterone, estrogen. So these will control internal biochemical processes. And these may limit the cell's processes in order for them to work, the limbic system or the reproductive system, or whatever. And even the cells themselves. Okay, so there are other types of lipids that we can go through. Chlorophylls and carotenoids. These are pigments that absorb light energy, help convert that into chemical energy. Those are lipids that we're definitely going to go through this semester. There's also lipids that combine with carbohydrates. Those are called glycolipids. They're ones that combine with proteins. Those are lipoproteins. And they have important structural and functional roles in cell membranes, cell development, things like that. Next, we're going to talk about proteins. Proteins in general are the workers of the cells. These are structural, these are enzymatic, these are transport, these help with DNA, these are hormonal, these are toxins, venoms, whatever. These are the workers of the cell. Okay? Proteins are macromolecules. Okay? These are made up of amino acids, right? They contain both amino and carboxyl groups put together. So you have 20 different types of amino acids to build these proteins. They have all of the same type of structural plan. You have a central carbon atom attached to an amino group and a carboxyl group, a hydrogen atom and a variable R group. Right? That's all of them, how they work. That's why they're called amino acids. Right? You have the H group, which is the acid part, and then you have the amino group, which is amino. Some side groups are polar and some are non-polar. Some are negatively charged, some are positively charged. You know, we can go through those. Many side groups contain functional groups, such as another amine, another hydroxyl, sulfhydryl groups can produce disulfide linkages, right? Help proteins control their 3D shape. So we have the non-polar amino acids. We have alanine, valine, leucine, isoleucine. You can go through these. Uncharged polar amino acids. I don't expect you to learn all these, but you should probably know that they exist and what they are. Negatively charged amino acids, aspartic acid and glutamic acid. Positively charged amino acids, we have lysine, arginine, and histidine. And then we have this disulfide linkage, right? So this disulfide linkage, if you have a sulfide and a sulfide bond on one side, if you have a cysteine amino group, that two sulfur atoms are going to covalently form a disulfide linkage. And sometimes that'll make the protein form in a way that otherwise wouldn't form. Okay? And it would make the three-dimensional form differently. Okay. Covalent peptide bonds, just the same as any, we go through dehydration synthesis, right? This is just called a peptide bond. Peptide bonds linked amino acids into polypeptide chains. The subunits of proteins goes through dehydration synthesis, links the carbon and nitrogen atoms together. The growing polypeptide chain has a nitrogen terminal end and a carbon terminal end, right? Which links them both together. The new amino acids are linked only to the carbon terminal end. Now, when we talk about protein structure, we have 4 different levels of protein structure. We have a primary structure, which is the basic unique sequence of the polypeptide. Right? We have the different amino acids. If we have methionine, phenylalanine, aspartame, alanine, arginine, all of them in chain. That's the primary structure. Then we talk about secondary structure. Secondary structure is produced by the twists and turns of that chain. Okay. If you say the primary structure is kind of like yarn, the secondary structure is kind of like you building the yarn into a quilt. So beta sheet is just like it going back and forth, and alpha helix is just like it twisting into a spiral. This is kind of like a fancy quilt. Tertiary structure is its folding. Okay, is its overall 3-dimensional shape. Okay, secondary structures, its overall 3-dimensional shape. So that's beta sheets and alpha helixes. All right. So if you have all the beta sheets and all the alpha helixes put together into one, that's the tertiary structure. Now, not all proteins are going to have this level, but you can have some that have a quaternary structure. Okay. Quaternary structures contain more than one polypeptide chain. Sometimes you have some that will have more than one polypeptide chain. So sometimes you have one monomer over here, one more over here, and then one more over here, and then one more over here, then one more over here. Sometimes they'll have like 8 to 10 to 50. It just depends on how many different polypeptide chains you need in one structure to make sure that the overall protein needs to function the way that it needs to function. I believe hemoglobin needs 4 polypeptide chains in order for it to function properly. Actually, that's what I'm going to have you do. For your attendance assignment, what I want you to do is name a protein in your body that needs a quaternary structure in order to function the way that it needs to do it. Okay, so go up on to some type of research. So go into just a sentence or something and just say in a sentence, tell me a protein in your body that needs more than one polypeptide chain in order to function. Protein properties and functions. Any type of process that unfolds the protein in order for it to lose its structure or lose its function is called denaturation. Right? And this can be caused by chemical, temperature, pH, anything that unfolds the protein. For some proteins, this is permanent. For others, this is reversible, and that's called renaturation. Guiding proteins. Certain proteins need a certain other type of protein in order for it to be guided into position. And this is called chaperone proteins, or chaperonins. Proteins link with lipids. again called lipoproteins. Proteins can link with carbohydrates called glycoproteins, which function as antibodies or receptor molecules for viruses. Proteins can link with nucleic acids. These are called nucleoproteins. These can form structures with chromosomes. Really quick, let's go through nucleotides and nucleic acids. So a nucleotide is a monomer of a nucleic acid. So the nucleic acid itself is the linked together nucleotides. So one nucleotide consists of a nitrogenous base, a 5-carbon sugar, that's a pentose, and 3 phosphate groups, or 1 to 3 phosphate groups, depending on what is all going on. Nucleic acids, you can have 2 different types. You have DNA and RNA, right? DNA is deoxyribonucleic acid. You have 1 less oxygen than RNA, which is ribonucleic acid, right? So DNA has 4 oxygens, RNA has 5 oxygens. So you're going to have the nitrogenous base, you're going to have the sugar, the 5-carbon sugar, ribose. It's going to be a pentose type of sugar, but it's ribose in ribonucleic acid and deoxyribose in deoxyribonucleic acid. Right? And then a phosphate group. 2 types of nitrogenous bases. You're going to have pyrimidines and purines. Okay, so the pyrimidines, you're going to have uracil, which comes only in the RNA, the thymine, and the cytosine. The thymine only comes in DNA, right? Uracil and thymine are interswitched. And then cytosine. And then purines are adenine and guanine, right? So pyrimidines only have 1 carbon nitrogen ring. Purines have 2 nitrogen or carbon nitrogen rings. The best way I think to figure this out is that all gold is pure, right? All gold is pure. Ag is pure. And then pyrimidines, I don't know the other ones. That's the best way that I figured it out, is just all gold is pure. So then you have DNA and RNA, how these consist of. So polynucleotide chains, right? You're going to have 1 nucleotide linked to the next by what's called a phosphodiester bond. Okay? So, diester. 1, 2, that's 2 diesters. It's having phospho linked to 1, 2 oxygens. So 1 nucleotide is linked to the next by bringing the phosphate group between the 5' carbon of one sugar and the 3' carbon of the next sugar. And that's always the way that it's going to go. It's always 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime. And I say this because that's always the way that things are run whenever you read DNA, 5 prime to 3 prime. Okay? Whenever we talk about DNA replication, whenever we talk about translation, transcription, it's always going to be 5 prime to 3 prime. Okay? And this, adenosine to thymine and guanine to cytosine, A to T, G to C. It's always going to be that way. Okay, so the bases are going to go A to T and G to C. They're always going to be bonded that way. Okay, likely. Keep in mind that there is mutation and this is biology. All right, A to T, C to G. That's the way that it's going to be naturally. That's it for this chapter. Keep in mind of the attendance assignment that I talked about. If you have any questions, please let me know. And I hope this was informative. All right, I will see you later.